So for our final speaker, we have Joergen, and he is a postdoctoral associate at MIT in the lab of Michael Fee. And he'll be talking to us about a connectomic substrate of credit assignment in basal ganglia reinforcement learning.
Thank you so much for this introduction. So let's start the presentation. This works, right? So yeah, I mean, here in the background, you can basically see already that I'll talk about a lot of data. So about a basal or several basal ganglia connectomes, and I basically want to see what we can learn from this like static data about credit assignment in reinforcement learning. So let me first talk about what I mean with credit assignment. Well, this is actually a very general problem, as you know. So basically, when you want to optimize the behavior of a system, then you have to find the relevant parameters that need to be changed, and you have to understand how to adjust these parameters. And a very successful solution for this problem is, as we all know, the error backpropagation algorithm, which is already pretty old, but incredibly successful with its many variants. But the big question for backpropagation is basically, is it actually implemented in the brain? And as you know, there's a lot of recent work, especially theoretical work in this regard. But I think we still don't have like an idea whether an animal actually uses this approach. But there is a second class of algorithms, and I call them here the perturbation-based approaches, which are usually used in combination with reinforcement learning, and they're like even simpler conceptually. So you just like wiggle one of the weights or parameters, and then you basically look at whether the output changes in a positive or negative way. And the big issue with with this class of algorithms, on the other hand, is that they're assumed to basically be too slow for learning, so that convergence for real learning problems is too slow. So I want to understand and study how the basal ganglia, which according at least to Kenji Doya is like the reinforcement learning machinery of vertebrates, solve this problem. And I think we actually have now reasonable evidence, especially in the songbird system that I like to study, that perturbation-based approaches are used for song learning. So when you look at how zebra finches learn their songs, so young zebra finches, they basically start out with very simple and unstructured song that we call subsong, and then over time they gradually refine this, so the song becomes more structured and more repetitive, because zebra finches have a very stereotyped and repetitive song, and then eventually you end up at a very stereotyped and but refined adult song. And what machinery do they use for this? Well, I talked already about the basal ganglia, and it turns out that in fact they have circuitry in their brain that involves the basal ganglia that is required and necessary to perform these this imitation learning here. And we have essentially here in the basal ganglia these medium spiny neurons, so they're filled up with medium spiny neurons, and then we have two different cortical areas that are projecting onto these medium spiny neurons. The area that is called HVC, and another area that is called LMAN, and then we have a third non-cortical area that is called VTA, that also projects there. So how do we think that credit assignment actually works there in the basal ganglia? And Michael Fee, my advisor, came already together with Jesse Goldberg already a few years ago up with a very interesting model in this regard, so that basically credit assignment combines action, copy, context, and reward signals. And when you look for these three signals in the songbird brain, well, then you can essentially find areas that almost perfectly match to these descriptions. So it turns out that HVC has these very statically relative to the behavior of firing neurons, so they can convey the relative song time. And then it turns out that LMAN, on the other hand, this other cortical area I just talked about, has more or less random activity when you watch the neurons fire during the behavior. And these random variations are also introducing little changes into the song. And this is, of course, exactly what we want for reinforcement learning, right? You want to have a behavior where you introduce small changes, and then you want to evaluate these changes and see whether they were actually positive for the animal or whether they were negative. And it turns out that there's this third projection that in just a few years ago, and Jesse Goldberg's lab, Vikram Gadagar, showed in a very nice paper that VTA is basically conveying a reward, almost classical reward prediction error into the basal ganglia area here, onto the medium spiny neurons. Okay, so now with these three signals together, the question is, and you rightfully may ask at this point, what does all of this have to do with the connectome and anatomy? And it's actually fairly straightforward. So we derived three key anatomical hypotheses based from this model. And the first one states that in this model, it makes a lot of sense that the HPC or context synapses are actually the synapses that are being modified and identified with this credit assignment mechanism. Why is that? Well, because they're static relative to the behavior and the activity. So if you want to learn something, you want to base it on a stable context, but it's impossible to base learning on something that just varies and fluctuates randomly. So we predict that essentially the HPC context synapses target the plasticity compartments of the medium spine neurons, the spines, but that the element synapses, which convey this information about the random change that just happened, to the dendritic shaft. There's a second prediction that comes out of this, which is that pre-post, so synapse pairs between the same axon and the same dendrite from HPC are correlated in size, which can be taken as an anatomical marker of synaptic plasticity, because when you think about Hebbian plasticity, then it basically predicts that neurons that synapses that are similar in their pre-post activity should be similar in size. And the third prediction that we came up with, which is a more recent development, is actually that the basal ganglia should filter out the random activations that are coming in from these L-man axons. And why is that? Because again, because the behavior should be biased by the context of a certain situation, but not by the random perturbations that are currently going on. Okay, so to address these questions, I've acquired two massive electron microscopy data sets of this brain area of area X, and had really help from hundreds of students in this analysis, because this is such a time-consuming process to analyze these data. But since this was still not sufficient, even with all this help, I was fortunate enough that I could team up with Google AI, so with the team of Laura and Jane there, and they developed these amazing flood-filling neural networks that were then used in my data for automated neuron reconstruction. And I was also teaming up with two undergrads back then in the lab to basically solve the other segmentation and reconstruction problems to be able to then automatically analyze this data, which is essentially the identification of the synaptic junctions, but also synaptic vesicles. And then you want to classify all the compartments of these neurons automatically. You don't want to do this by hand, and of course, the cell types. Okay, so with all of this, I could then essentially access these connectomes and basically query them for these hypotheses that I've just laid out. So I can do these virtual experiments, and just to give you an overview of how much about how much data, actual measured connectivity data I'm talking about. So the small data set I've acquired contains already roughly 400 medium spiny neurons and over 250,000 synapses. But the larger data set is actually one of the largest reconstructed EM data sets in the world at the moment. It's not fully proofread, but even without the proofreading, we think it's extremely useful, and it contains now roughly 20 times the number of neurons, so over 4.5 million of synapses. So that's a lot of data to test the predictions. And to do this, I can now basically, so we wrote this little Python interface, and I can just here is 2 lines of code that give you an example. So give me give me all the HBC synapses in the data set, essentially when the cell type is HBC, and then I can calculate the mean size, for example, but only for those HBC synapses that actually target the medium spiny neurons and where the morphology on the postsynaptic side are dendritic shafts. Okay, so with this tool, I can now directly address these predictions I've talked about earlier. So the test whether HBC terminates on dendritic spines and whether the axons, so the context synapses terminate on dendritic spines and the action synapses on dendritic shafts. And it turns out that this is true. So the vast majority of HBC synapses terminated on MSN dendritic spines as predicted by the model, while there's a slight majority for the Elman synapses. And I can then could directly test the next prediction as well, so that synapses between the same axon and the same dendrite are correlated in size, but that this is the case especially for HBC, but not necessarily for Elman. And it turns out that there's a highly significant effect for HBC. It's relatively weak when you compare it, for example, to the same observations in hippocampus, which is interesting, but it's about similar effect size as in that is barrel cortex. Interestingly, this turns out to be not significant for shaft synapses at all. Okay, but now let's get to the third prediction. The third prediction was essentially that there's this feed-forward inhibition, and this is now a second-order connectivity analysis, but of course, it's very easy when you have this connectome available to just like search the connectivity matrix for these motifs. So this is what I did, and you can directly identify these Elman axons that are projecting onto interneurons, onto local interneurons, also onto medium spiny neurons, as we know. And then these interneurons project again onto the medium spiny neurons. So you get these triplets. And then on these triplets, I can now do quantitative anatomy. And so one of the first findings was, you may be aware of a recent study from Princeton, where they basically showed bimodal synapse size distributions for intracortical neurons. And it turns out that there are also bimodal synapse size distributions in the basal ganglia, but only for some connection types. So this is very important because it means that maybe something is special about these connections, and we're still like working on understanding what this could be. But I can also use these identified triplet motifs now to perform like more quantitative anatomy, for example, in terms of dendritic location of these synapses and whether they could actually be suitable to cancel out spikes. And this is what we find. So the inhibitory neurons are enriched here close to the soma, while the excitatory synapses are more far out. Okay, and with this, I already come to the last part. So we also did, based on these findings, we also did a small modeling study, like very simple firing rate models, actually with a local learning rule and eligibility trace, as Haran had like introduced in the beginning. And when you implement this model inspired by our anatomical findings, then you see that in a relatively small number of iterations, so less than 1,000, which is well within the number of repetitions that actually zebra finches use to learn their song, that you can copy a simple one-dimensional template. So with this, I'm already at the conclusions. So I think that EM connectome analysis is really a new, right? And new because it's actually the technology and theory is there since many years, but it was very difficult because of the technical challenges. A tool to test functional circuit models and ideas. And for this specific model here, to me, it looks like the data and simulations are consistent with this plausible, biologically plausible credit assignment mechanism that is based on converging action context and reward signals. And if you have, like, if you want to read up on this, then most of these results are already available as a preprint. Thanks.
Awesome. And, thanks so much, Joergen.
Yeah.
Sorry. So yeah, I know we're running pretty short on time here, but if anyone has like a quick question, I'd be happy to ask it. thought someone raised their hand, but maybe not. Okay.
Okay.
Yeah, I guess just a quick thing. I guess like how simplified do you have to be when you do the when you get these EM partitions, I guess, of the neurons, and they connect to a bunch of things, bunch of different neurons. How simplified do you have to be in terms of how you interpret the findings after that? I guess if that makes sense. Because I imagine there's a lot more complexity going on in there than what you're extracting.
Yeah, I mean, so the results that you saw, they were extremely simplified and targeted, right? I mean, so when you so I think there are in general two different ways of dealing with this kind of data. So the first one would be that you go in with specific predictions and just like almost exclusively look at what your model essentially says. And the other way is that you take a more explorative approach and just look at like all the different cell types and how they connect. And so in this case, I've taken like this like very focused approach, which can make your life much easier, right? Because of the amount of data, and there is some. I mean, there's some. So I'm not sure I fully understand the simplified aspect of the question, but I mean, the data is like not really simplified, right? So we are measuring synaptic areas for the different cell types, and then look at where they end up on the dendrite. So we have like very detailed anatomy and theory available, but we are not like using all of this anatomy all the time. Yeah, it really depends on the question.
Yeah, I guess it helps when you have a very focused